All right, so with this slide set, being comfortable with the endosymbiosis theory is a good idea. And then I'm not really going to, say what year, how many millions of years ago did such and such evolve? That's not really how I would do it. It would be more like, do you understand the order of who came first, who's next, who's next? But memorizing like how many millions of years ago, that information shifts all the time. So that's not incredibly helpful. It's somewhat helpful. I'm more concerned that you understand the progression of who got to land first, the bryophytes, those lovely mosses, liverworts, and hornworts, and then, then what? Who came after that? And then one of the other ideas that really, if you're thinking like a plant evolutionary biologist, you like slide 13, what's differentiating? What did they do? Each group of plants kind of did their own spin on, well, this is what we're going to do to evolve and survive. And so it's like, okay, well, what is that? The bryophytes ended up being able to protect their embryos much better than the algae that, they derive from. Whereas the seedless vascular plants that went, oh yeah, well, we're going to do vascular tissue, and that way we can spread onto drier habitats. We don't have to stay in a moist habitat. And then the gymnosperms came along and said, oh yeah, well, we're going to one up you. We're going to make a seed instead of using spores as a unit of dispersal. And so these types of ideas, these are it's a big deal to think about, what's going on in terms of the innovation, the adaptation, the evolutionary strategies they had, and what advantage that gave them. So that definitely I'll be asking about. And believe it or not, I'm going to ask you to identify flower parts. It's going to come into play off and on through the semester. So let's get comfortable with it. I don't care about the peduncle. Remember that? You don't need to know that. But like this part, the peers ants, was like this stuff. I don't really care about, but the other stuff you should feel comfortable with. For peduncle. Yeah, for peduncle, it's always left out. Okay. Questions on any of the concepts in this or need clarification on anything in here?
For the endosymbiosis theory, like just kind of knowing general.
Yeah, we didn't go into huge detail, but what makes a plant is that additional chloroplast endosymbiosis event. Wouldn't that be nice? Have chloroplasts. Hey, I just discovered that there's a sea slug that has chloroplasts. I'm like, what? How come we don't have those? If a sea slug can have them, come on. I bet someday they'll figure out some way to do that. Genetically modify us. Anyways, anything else on here? Questions? All right, what did we do next? Plant microbes stuff next. So the plant microbe stuff, think of this as kind of like an intro. This idea of plants and their associated microbes will come back through the semester. So this was more like, let's set the stage here for what we got going on with plants and their microbes. But there's a lot of big concepts here to think about. And some of these concepts go with some of the readings. So so basically, thinking about the plant body above ground and below ground. Below ground is really incredibly prolific in terms of their interaction, much more so than above ground. Because the soil is just chock full of microbes. And microbes love to hang out with plant roots. Plant roots provide a lot of nutrition for those microbes. Um, let's see. So we thought a little bit about the plant body and how it's well colonized. And then we talked a little bit about the mycorrhizal fungi. So with the slide sets, so like some of the readings talk about mycorrhizal fungi and all of that. But when you're looking at these slide sets, I will actually be pulling some information and asking questions from what we talked about off these slides. So for example, the pie chart, we talked about who's dominating in terms of the number of plants that have certain mycorrhizae. And then we did a comparison of, we zoomed in on the ectomycorrhizae and the VAM or endomycorrhizae, and then the orchid mycorrhizae. So I'll ask you specific questions about those, like what are their characteristics? How are they similar? How are they different? All right, so when you're studying these slide sets, you will get specific information asked about them. Sometimes it's like one right answer, and sometimes you have wiggle room, like give me something, that kind of idea. So if you're unsure when you're answering any of these questions on Wednesday, that's why I want to be here so that you can ask, is this what you're looking for? Is this what you mean? And I can give you feedback. Because some of the questions that are more open-ended, or like there's more than one way to answer it, sometimes that can feel a little weird to students taking the exam. It's like, well, is this really what she wants or not? And so make sure to ask questions. And of course, ask questions on anything that's confusingly worded. I will try to rephrase it. Certainly, we talked about endophytes. So think about the material, the concepts covered here, like what are they? How are they interacting with the plant? And of course, the PGPRs or PGPMs. Questions on any of those groups of organisms?
You said everything's gonna be like written, right? Or like.
Oh, good.
some, like,
So I said last week, no multiple choice. I lied. There's one, I think one so far, and then there's matching. Oh, I put matching in. Couple matching questions, so there'll be like a word bank. So you'll have that, but it's minority. Most of it's short essay or long essay and short answer.
The
And
best way to go through all this is just like practice like writing out
Mm.
the concept pretty
Mhm.
much.
Yeah, I would strongly put, as you're studying, put yourself in a exam scenario. Like, can I do this? Like, without looking at my notes after I've gone through it, I'm not going to look at my notes, and now I'm going to just type it out, see if I can come up with it. Because that's essentially what you'll be doing on Wednesday. So. The beauty of essay is it's partial credit, which is always nice. I'm always very optimistic. I'm like cheering for you, like, give me something. So it's nice to get a partial credit at the very least. And whole credit, of course, is best, but multiple choice, it's all or nothing, which is kind of a downer. Yes.
For the shorter essay questions, how many points are those ones with?
So usually when I say short essay, those are usually around 4 points-ish, 4 to 6 points. And then longer ones are 10 and above typically, where I'm asking for more, give me more, like more than a few sentences. Or the concept is particularly intricate and it requires some good explaining, that type of idea. So look at the point value of each question if that helps you to get a feel for how much I'm looking for, and ask, I've had, if you want to like type up your answer and then say, is this what you're looking for? if there's something missing, I'll say, well, and I'll try to tilt you in the right direction. But so yeah, that's it's okay to type it out and then say, is this enough or do you want more? Other questions on this slide set or? We went into the soil as well a little bit, like just kind of some of the general characteristics of the soil that you need to understand for this class. So obviously I'll be asking about that. So much of how a plant interacts with its microbes, it's, soil, that's where it's at. And so understanding the soil environment is a good idea. The basics anyway. I will ask about the nitroplast. I'll flatly tell you that. That's such a cool discovery. I'd like you to know that because that's a huge breakthrough that they discovered. So that's the first time I've ever got to teach that. It's so new. So if you've taken plant phys with me, yes, I'll ask about nitrogen fixation, but not nearly to the level we do in plant phys. We don't need this isn't a physiology class, so we're not getting into the details. However, still need to know the basics. Should we move on to the next slide set? So the next one is plant signaling and plant neurobiology signaling behavior. That's a big slide set. So no, I'm not going to ask you to explain how our brain works because I don't even know fully. It's pretty amazing. But the very basics, I think that video can be helpful on how the human brain works. I tried to capture some of the content from the video and put it into the PowerPoint to get you thinking about, this section in particular. Think about these ideas of, the synapses, the neurotransmitters, the vesicles that hold the neurotransmitters, the calcium, so the very basics because that's what they are looking at when they go into the plant world. Obviously, plants don't have brains, but they do have calcium flow. They do have vesicle trafficking. They do have their version of neurotransmitters, some of the same ones that we have. GABA has been confirmed. Glutamate has been confirmed to be in plants as well, and it seems to be acting as a way to transmit electrical information. So no, they don't have brains, but they have their analogous at least the ability to transport, communicate through their body with electrical impulses. So what questions do you have on this slide set? So it looks, the first, I don't know, 24 slides is really looking more at the kind of the neurobiology idea and where they're looking for evidence of neurotransmitter-like molecules. So it kind of splits there and then we start getting into, oh yeah, I think that idea of using anesthetics as a way to show, I thought that was really interesting. So make sure to look at that. They point to that when they look at animals and other organisms as kind of data to show that there is something going on with electrical impulses providing information. But then we got into movements. How do plants move? Definitely need to think about that too. No, I guess the neurobiology goes beyond slide 24. We're all the way. Oh yeah, memory. So we had to get into that too, thinking about that kind of activity. It'd be a good idea to know the definition of memory. I wouldn't be surprised if that shows up as just give me the definition. The broadly defined definition, not how we do memory, but the broad definition. So this, like I could be phrasing some of this content as, give me some examples of memory in plants. So you wouldn't have to spit all this back at me, but give me some examples from this. I would like you to be familiar with her experiment with the mimosa. Like, can you explain the basic setup and the basic results, which is pretty much on the slide set, but the paper's posted as well, and some of their conclusions. I just read Monica Gagliano, who did this research. She has a $1 million grant currently and she got it from a nonprofit, not a government organization. So her funding didn't get cut with all the budget cuts, the government cuts. So and so she's not beholden to the same system as most scientists. When you write a grant, it's usually to governmental agencies and you have to follow a very strict, it's like a contract. This is what I propose to do and you have to do it. And she'll have more luxury to play around a little bit, which suits her really well because that's kind of how she does her research. She plays before she starts getting down into a hypothesis. So yeah, $1 million. Good on her. So I may, refer to her paper, like the Mimosa paper with Monica Gagliano, and then I may ask about like the Thilo Henning's paper on the pollination, bumblebee pollination. So start learning it that way, like understand what I'm talking about. If I phrase it that way, know what ones I'm zooming into here. And then thinking about how plants move, that's important. That's going to come up again through the semester. How are they doing that? So we looked at the turgor or the growth, and then the sigmomorpho is just kind of an extra example of how touching them can change their morphology so greatly. And we looked a little bit at smell. We'll do more smell later in the semester too. But, I may ask you about the bean plant with the so the slide 86 where the experimental setup, thinking about how they set it up. And, they are monitoring not only the chemical shift going on in the leaves, but also the VOCs coming off of the attacked leaf. And then they had to cover the leaf to do more experimentation covering that leaf all with that plastic bag to show that it really is a VOC impact. And again, I'm not so much like, it's not like I'm not really asking you to memorize specific hormones like slide 90, like what we do in plant phys. It's more like you understand the concept, like some hormones can go volatile. That's more what I'm going for. You don't have to necessarily know jasmonic acid goes volatile or salicylic acid goes volatile. It's more of, in general, we know some hormones can go systemic and then some can turn into their volatile form and do that. Questions on this slide set? All right, the last slide set. Transgenerational epigenetics. That's a pretty short one. Again, kind of the basic ideas. There's so much to this, but and one thing I forgot to put in the slide set, I should have put in here, microbes can be epigenetic to the plant. So if you go back, I should have put this slide in here again and I forgot to. Where is it? This slide in plant microbe. This slide here, where it shows the microbes in the roots messing with the plant's gene expression. That's an epigenetic thing. And I forgot to put that in. I don't know why I forgot to put that in, but I did. So, anyways, going back to the epigenetics, some of the basic ideas, know the definition. There's a couple different ways you can define it. Depends on the wording. I like the shortest one. Change in phenotype without a change in genotype. Boom, you got it. And basics of DNA, the histones that you can get alterations there as well as actual DNA strands. The fruit fly experiment, I think that one is nice and it really did a good example of and that did involve the microbes in the gut of the fruit fly. So I guess at least you got some of the microbe influence there as an epigenetic impact. But this gets so complicated so quickly. I just gave you some very basic ideas. And like, what have they found? Like, what is epigenetics doing for the plant? That last slide. So a lot of the very important stuff that a plant really cares about, like flowering and how to adapt to cold and biotic and abiotic stress in general. So it seems to be a pretty heavily used, which makes sense again when you think about plants being their lifestyle, they're so stationary, that doing epigenetic mechanisms makes a whole lot of sense. But we're not a genetics class, so we're not going to get too deep into it. Questions on epigenetics slide set? Then, so those are the slide sets. Then we also have the readings and videos, and those go hand in hand with what we were covering in class together with these slide sets. But do make sure that you look to those to support what some of the concepts are in the slide set. I really didn't get into talking about that video, what plants talk about documentary. I didn't really get into that in any specific way, I guess, but it does support a lot of what we're talking about with behavior of plants. But please watch that so you can give some specific examples from it. There's a ton of cool examples in there. It's a really good documentary. So make sure that you do think about that and are capable of giving me some ideas on that. And then we had that holobiont reading and the plant microbe reading. And then several videos that are more for just to help you understand. I obviously, like I said, I'm not going to ask you about how our brain works in any detail, but some of those videos can be helpful. Oh, and then there's that one by, what's that one? Um, The Roots of Plant Intelligence. And then the electrical experiments like that, he goes over with mimosa and the Venus flytrap. So some of that information can be really helpful for you to give examples when I ask you an essay question. You know, you could refer to what you learned out of that video, and same with the Roots of Plant Intelligence. So when you're asked to give an example, sometimes it's easier to remember something from a video than reading and studying slides. So I'm trying to find useful videos in case you learn that way. Some people like to learn that way. Okay. And then the what plants talk about is on there too. What else? What other questions? Oh, so point values, kind of the plant signaling and behavior. Is that what I call it? The PowerPoint slide. Yeah, plant signaling behavior. It's a big slide set. A lot of points are coming from that. That's a big topic. Lots of topics in there, I guess I should say. So if you're kind of breaking it down that way, you'll get more points on those topics than the other topics, for sure. But plant microbes, fair bit of points coming from that too. But probably the majority of exam points will be coming from thinking about that slide set and the associated readings on how plants signaling and behavior. So do we have power outlets in here? Like there's no power outlets per table, is there? There's nothing
There's some, these doors,
on the floor.
yeah,
Do they actually work?
think so.
And then there's wall. So, you know, if you need to plug in, you can feel free to get up and move around quietly if you don't want to mess with. I've heard some rooms, the floor ones don't work, but maybe they fixed them. That was years ago that I heard that, so hopefully they fixed them by now. But bring your power cord if you don't have a very good battery, because you'll have the full lecture session, okay? And make sure to ask questions. And I gotta finish making the exam before I can give it to you. I got homework. It's agonizing. My problem is what I end up doing is the first draft exam is way long. And then I gotta go and cut back, because everything's so interesting. But then I gotta cut back. So that's kind of the stage I'm in now. It's like, oh, I gotta cut back. So, all right, we're done. Move on to your next adventure.